And this whole set of books together is the human genome. Inside every single cell, you have every instruction you would ever need to accomplish any bodily task that you have to, do every day. But, your genome is yours. Your set of instructions would be pretty useless to a cat, right? You have different needs on a day-to-day basis. So genomes differ from species to species. And beyond that, even our own genomes change over time. Okay, so the whole set of instructions you might have needed to survive when you were, young or living ages ago might be very different from your needs at the present moment when we're living longer. And so that's one reason why scientists have been trying to study just exactly what is there. And if there are changes, what are the effects of those changes? And so one change of a letter in this DNA genetic code can really change a lot. So let's say I replace this A here with a T. Does everybody see that here? I replace the A with a T, okay? Basically, one letter can change a lot. Let's say I have this whole book, right? How many nucleotides in the human genome? I said, 3 billion letters. If you were writing a book that's 3 billion letters long, what are the chances that you're going to make a mistake? They're pretty substantial, right? There's a chance that something will go wrong. We're not perfect, right? Sometimes these things happen. So let's say that you have a change in one of the nucleotides in your genome. Basically, what's going to happen then is you're going to change the instructions, and that's going to alter the structure of that protein you make, right? If you have a manual that instructs you how to build something, let's say you want to build a lawnmower, and a page is ripped out, or there's a typo, you might not have the same set of instructions. It's going to look pretty different. And so that's basically what I'm trying to get at there. And when you have a change like this in the genome, a change of a nucleotide for a different letter in the genome, genetic code, we call that a single nucleotide polymorphism, or a SNP. And so one letter can change a lot. I showed you the example before is what happens to hemoglobin if you change a letter. So in sickle cell anemia, if you change an A in the hemoglobin gene to a T, that makes the hemoglobin not form correctly and it clumps together. And that makes your red blood cells, which carry oxygen, kind of collapse and look like this. And this is an example of a single gene disorder, which is a change in one nucleotide in the genome that causes a disease, sickle cell anemia. But SNPs don't always cause disease. We know we have variations in our genome that just make us different, like eye color, right? Depending on which SNPs you inherit from mom and dad, you're going to have a different set of instructions for how to make the color in your eyes. That's why people have such varying degrees of eye color. But basically, if a genome is 3 billion nucleotides long, and we have these SNPs in roughly about 1% of our genome that we know today, maybe like 1 million SNPs or so, there are a lot of different combinations, and that's what makes us all unique individuals. And that's one reason why people tried to pioneer the Human Genome Project 2 decades ago, right? So here you see former President Bill Clinton. He's standing with Francis Collins holding that book there. He's currently the director of the NIH. And to his left is J. Kirk Bender, who led the private-funded part of this partnership to sequence the human genome. And basically the reason people wanted to do this was to see what the heck is in all of these cells? How many genes are there? What are they doing? How many changes are there? How do they contribute to disease? So we did this. It took thousands of people. It took 20 years, it took billions of dollars, and we did that. But today we know that we've only looked at a very small number of human genomes, and we have billions of people on the planet. There's a lot more variation than what we've captured. And that's the reason why today, 10 years after the completion of the Human Genome Project, we're looking not at one human genome, but we're looking to sequence 1,000 genomes to really try to get a sense of how much variation there is. And the reason that's possible today is because the cost of sequencing, the cost of the technology that we use to look at the human genome has dropped a lot over time. So if you look here, do we have a laser pointer? Yes. If you look over here, this is showing how much money it costs to look at a human genome starting in 1990 all the way to the present day here. And so 20 years ago, it cost $3 billion to sequence a human genome. Today, it costs around $1,000. So it's dropped a lot in price. However, for the average American today, I'm sure $1,000 is still more than you want to shell out for a genetic test. And so, for that reason, there are companies that exist now that look not at your entire genome, but just that 1% that we know has all these SNPs. So for $100, roughly, you can write to one of these companies, like this one here called 23andMe, you can spit in a tube, send it to them, and they will actually look at your DNA and look at to see which SNPs you have. And just from looking at these SNPs, they can tell you your genetic ancestry, where your founding family members came from. They can tell you if you carry any single gene disorders like sickle cell anemia. They can, using current studies to date, predict your risk for things like polygenic disorders or complex diseases like diabetes or heart disease or cancers, and they can give you some percentage of risk for those diseases. And they can also look at other traits like drug response or features. So I actually, being a geneticist myself, was really tempted by this offer. A few years ago it was around $500, but I bought a kit on DNA Day when it was a $99 sales special because I'm cheap. And, and basically I did it myself. And what you get when you do a test like this is a report of a whole lot of traits. So up here you have your single gene disorders. Thankfully I don't carry any of those. Over here you have disease risk predictions for some complex diseases. You can see I have a 5% risk of restless leg syndrome, one of the less scary things in this report. You can see things like certain traits like if I flush if I drink alcohol or not, I likely have brown eyes. And you can see things like drug response. So one thing I might want to point out is, why is it a percentage and not a diagnosis, right? And so the reason for that is because your genome doesn't exist in a bubble. You're constantly interacting with your environment, and the environment plays a big part in how your genes behave and what signals they send to your cells and the rest of your body. And beyond just the environmental influences that you have on a day-to-day basis, you also have to take into account that all of the reports you get in a test like this are actually compilations of research that other scientists have done where they looked at, oh, here's a group of people with the disease, here's a group of people without the disease, let's see if there are any SNPs that differ between the two groups, right? And so depending on which source population they use, they might get a different outcome. And so that's why you get a risk percentage and not a clear-cut diagnosis like you do with single gene disorders, okay? Because all these genes in different combinations could be working together, not just alone. The other thing you might want to consider is things like drug response, which tell you not only about diagnosis and prevention, but also treatment options, just based on DNA, right? So this is drug warfarin, otherwise known as Coumadin, and basically if you ever have a stroke, they might give you this. It's a blood thinner to prevent your chances of having a blood clot. But how you break down this drug is actually determined by your genes. So some people carry SNPs in their genes that make them break down the drug really, really quickly, and so they don't really need a lot of it. Other people break down the drug really slowly, and if they break down the drug really slowly, they don't need a lot. It'll last them a while. If they go through it really quickly, then they need a low dose, or it'll go all over the place at once. And basically, if you have too high a dose of warfarin, then you have hemorrhage and you die. So it's really bad. So it's good to know that if I ever have, risk of blood clot, that I should only need a little bit of warfarin and not too much. Presently, doctors wouldn't be able to do something like this, right? The way that people decide which dose of warfarin to prescribe a patient now is based on race. As a rough proxy for what gene you might carry. And it's an imperfect system, and I'll show you why in a minute. The other thing we can get with these SNPs is genetic ancestry, because as I said, we come from different places over time, different genes are inherited from mom and dad all over the world, and you can tell based on which SNPs exist in a population where that population is from. So over time, what you're looking at, these are a list of chromosomes here, okay? I told you, remember, there are 23 of them. Half of it comes from mom, half comes from dad. And you can see each color represents a different ancestry. So blue is European, green is African, and orange is Asian, or sometimes maybe Native American. And so basically, this can give you a much more accurate information about what your ancestry is. Because race itself isn't necessarily important for medicine, but what is important is which genes you've inherited. So for example, if you're this guy, and you walk into a hospital, and you don't know that he's the president of the United States and has a white mother and a black father, you might say he's African-American, give him a high dose of warfarin because African-Americans are slow warfarin metabolizers, and so that might actually not be a good idea because since he has some SNPs for the European version of his gene, he might be too sensitive to warfarin. But if you just looked at him, you wouldn't know that. So in that sense, looking at your genome can help you have the right treatments at the right dosage. So you're not only minimizing risk for developing diseases, you're maximizing the treatability of those conditions. One thing I do want to point out, I don't know if you can see, does anyone here have curly hair? Can I have a volunteer for a second? Is anyone brave? Come here. Okay. I've always wanted to do this. Can you just, here. Okay, so my hair won't bounce. I definitely don't have curly hair, right? Thank you, Jessica. So how could this happen? How could this, genetic test that I spit in that seems to be pretty accurate for all these things tell me that I have curly hair when clearly I don't? And again, this goes back to the fact that all of the reports you see here are based on other studies that were done. If you actually look to see this study, because with every trait you see here, you can click on a link and it'll tell you which scientific study that finding occurred in, which SNP they looked at, which population they looked at. And when you're doing these studies, comparing groups of people with a trait and without a trait, it's really, really important to match age and sex and race, because otherwise you might be ascribing differences to genetics when it's actually due to a lot of environmental components. And so that's why this is actually not accurate. So you should take these things with a grain of salt. You shouldn't look at them by yourself. There's a lot of environmental influences. And so that's why I really want to stress to you, your genome is not a death sentence. There might be some things in there that look kind of scary, but again, it's a percentage of risk, and a lot of that risk can be reduced with behavior. And that's one reason why in 2008, George W. Bush passed the Genetic Discrimination Act, which actually protects you from discrimination based on DNA differences as far as access to health insurance and access to employment. Because nothing is set in stone. Just because you carry a certain variant for a gene doesn't necessarily mean you're going to develop it. There might be a risk, but that's no reason for someone to deny you opportunity. However, on that note, I would like to stress that your genome, like your health records, are private, and you shouldn't go around putting them on Facebook and things like that. I shared with you this screen, but if you notice, I haven't given you any actual genotypes or anything because that's kind of my own private deal. And so I'm happy to talk about it with you guys, but just make sure that, this is you wouldn't want to put the results of colonoscopy on Facebook, maybe you would. But if you're into that, but I personally think that it's really important that this is your information for you and your doctor to discuss. And so basically the goal of moving genomics forward is to work with your doctor so that you can find ways to really make medicine as effective as possible for you. And one other thing I want to stress, to make sure that things like this don't happen, is participate. There are genetic studies happening every day. The only way that you're going to really find if a finding is relevant to you is by having your population be represented in the studies. And so a lot of these studies focus on European Americans, some of them focus on Asians. A lot of the genetic studies haven't studied African Americans, so there's a whole lot of variation out there that we don't know how it contributes to the disease. And we won't know until these people start participating in these studies. So we have a better idea of how relevant each SNP is for your disease risk. So basically I told you your genome holds a lot of information. It's tiny. You can use it for diagnosis, you can use it to predict disease, you can use it to prevent disease, and you can use it to treat diseases. And on that note, I'm going to pass it over to Yisha.